Thank you for the introduction, and thank you everyone for joining this webinar. I'm really happy to tell you some exciting things about mass photometry. This work was done in Oxford University when I was a postdoc in Philip Kukura's lab. And today I'm going to tell you about a new tool to quantify your protein samples. So if we start with the motivation, most of protein studies start with extracting your protein or expressing and then extracting your protein from live tissues. Then you work really hard to purify your protein. Here the optimization of your purification protocol will have tremendous effect on the quality of your study. After purification, you find the best conditions for your study. You screen for different conditions. And then you do your protein study, and it can range from structural studies where you use cryo-EM or X-ray crystallography, and all the way to functional studies and biochemical studies. Ideally, you would like to know the content of your protein sample both during the purification, so you can optimize your purification protocol in the most efficient way, and when you screen for your best conditions for your protein study, and also even during your study. And therefore, a handful of techniques were developed to address this need, where ideally you will get an information which is in high resolution with minimal consumption of your protein and of course high throughput, so fast. If we look at the standard tools in protein sample characterization, we can roughly divide it into two types. One is bulk measurement and the other is single particle measurement. And examples for bulk measurements are SDS gels, chromatography, and DLS, where all those techniques are usually quite fast and simple, and they have the advantage of being available in most of the biochemical labs. The disadvantage is that it's low resolution, so getting a high resolution mass distribution is usually not possible. And if we look at the timescales for getting a result, it will take around a minute to a few hours. Now, with single particle measurement, an example here is negative staining. Here, this technique is much more detailed and accurate, and it has the advantage of providing some structural information about your protein. The main disadvantage is that it's usually too slow for efficient and high throughput protocol optimization. So if you really want to quantify the content of your sample, it will take between days to weeks. So is it possible to have a tool that will be both high resolution and high throughput? And we ask basically, can mass photometry accurately provide information on sample purity and heterogeneity in high resolution and high throughput? And of course, the answer is yes. And our goal was to show it in a quantitative way for different types of complexes and comparing it to standard tools, and highlighting its benefits and its limitations. All the results I'm going to show you today were published recently in Nature Communication, and here below you can see the reference. So let's start with an introduction, or what is mass photometry. So mass photometry is based on a technique called interferometric scattering microscopy, or ISCAT. And the idea is really simple. The idea is to take the interference between a scattering light and reflected light. So if you have a microscope and you shine light on the sample. The sample, most of the light goes through, some of the light is reflected, some of the light is scattered from a scattering object in the sample. You collect all the light that comes back from the sample, and then you image it on the camera. And if we look here on the sample cell, you can see different proteins. Those proteins come to the interface between glass and water. They bind in a non-specific way, and once they bind, they are scattered. Of course, we have also the reflected light that comes back from the interface between glass and water, and together they create the image that we see on the camera. And here to the right, you can see the equation of interference, where this is what we see on the camera, the combination of scattering light and reflected light. And usually, for small scatterers, the scattering amplitude, which is noted here as S, is really small, and you can neglect the pure scattering signal in this equation. Now R is the reflectivity, which is this term, and you have also here the interference between the scattering and the reflectivity. We can define ISCAT signal or ISCAT contrast as 1 minus the scattering amplitude divided by the reflectivity with a phase contribution. And the important, I think, thing is that the scattering amplitude is proportional to the volume of your particle. And if we think about most of protein samples, most of them has similar densities. So basically, the scattering signal is proportional to the mass. And this is quite strong, and theoretically, it was already realized several years ago, but it took a decade of optimization of both the hardware and the software to really make it possible experimentally. So what do we actually see when we look at the images? If you see here, this is the images that we see, or the raw images. And if you look here, it seems quite boring. And the reason is that there is a strong reflectivity from the glass-water interface, and this prevents us from looking at really the interference part. So we have to do some image analysis to see the signal. So before I explain what exactly we do, I want to show you the movie afterwards, after we do the image analysis. And here it is. So it's the same movie, only here what we see is what we call ratiometric movie or ratiometric imaging, where we take out the background and therefore we can see the signal. And here what you see is particles or proteins land on the interface. They create a signal and then they disappear. So what we do is ratiometric imaging, where we average both frames before and after a certain frame, and then we divide between the two. The before is considered the background, the after is considered the frame, and the particle signal increases and then decreases as we slide this averaging window throughout the whole movie. And here is an example of a particle which is contrast is increasing and then decreasing. And the maximum signal is the ISCAT contrast, which is proportional to mass. And this is exactly what we fit. And so it's important to understand that when the particle disappears, it doesn't mean that it's unbinding. It's basically becoming part of the background, and therefore it's removed. So as I told you, we fit the maximum signal, and here is an example of the accuracy of our fitting protocol, where here you can see for both high SNR of 12 and signal to noise and small signal to noise of 3.5, where the raw signal and the fitting fit quite nicely and the residue is within the fluctuations of the image. And then we fit each particle that we see in the movie and we create a histogram of contrast signals. And here's an example of contrast signal versus counts. And you can see different populations of particles. Here it's, of course, it's a protein. And you can see high contrast and low contrast, and it's quite nicely resolvable. So the only missing part is to convert the contrast to mass. And this is done by standard proteins where you know exactly the mass of the proteins. So here are two examples of calibrant. One is a protein that has different oligomeric state at 90, 180, 360, and 540 kilodalton. It's a monomer, dimer, tetramer, and hexamer. And here a second calibrant, which is a monomer at 66, dimer at 132, and a tetramer at 198 kilodalton. So once we know exactly the masses and the contrast, we can put it on one graph. And here you can see our calibration graph of contrast versus mass. This is known mass. And you can see quite nicely that it fits a linear curve with only a few percentage of error. So just to convince you that mass photometry is a really simple technique, here's a movie of how it works. Here is the instrument, and here you add a sample. Here it's to a gasket. You close the lid, and you already see your protein lands. You wait until the autofocus reach its position, and then you start record. The recording is usually between a minute or two, just to collect enough statistics. And then you do your data analysis. So basically it's, I would say, 2 minutes of a movie, and then another 1 minute or 2 minutes of image analysis. And then you have your at least contrast histogram. And then you apply the calibration and you have your mass histogram. So within 5 minutes, you can quantify your sample. So let's summarize. The idea behind mass photometry is that you have particles, proteins in our case, in your sample cell. They come to the surface and bind in a non-specific way, and they scatter light. The reflected light from the interface between water and glass will also interact with the scattering light and produce an interference, and this is what we see and collect. We look at the maximum signal and we fit it and we create a histogram. Here is an example of an experimental mass histogram and scattered plot of contrast versus time during the movie, where we collect all those dots. All those dots are fitting of the maximum signal, and we create this mass histogram. Here's an example of the signal, and you can see how the intensity of the spot increases as the mass increases. So if we summarize its main benefits, mass photometry is label-free, so you don't have to do any manipulation to your protein. It's a label-free single particle detection. It's in solution, so it's native-like measurement. It provides you with accurate mass measurement, including subpopulations. It's quick and simple, so you can get results within minutes, and it requires minimal sample. So an example is around 10 microliters of between 100 picomolar to 100 nanomolar. So in order to achieve our goal of showing that mass photometry can really be beneficial for protein sample characterization, we chose different biological systems. Each system will highlight a different aspect of the advantages of mass photometry. The first system is Anaphase promoting complex, where it's overexpressed in cells, followed by a multi-step purification to enrich the full complex abundance. And here we will show how we followed the full purification and we could really detect which sample is better and how it can be used to prioritize and optimize your protocols. The second system is Proteasome, where it's endogenous purification, so each preparation provides you completely different ratios between its subcomplexes, and then you want to know exactly what is your starting point in each preparation. And we will highlight also some extra benefits of mass photometry, specifically in the Proteasome. The third system is nuclear pore complex, where here we will illustrate how mass photometry can help cross-linking protocol optimization. The fourth system is Respiratory Complex I, where we show that mass photometry can also work with membrane proteins. And the last system is Cohesin, which is flexible protein, and we will show how mass photometry works with flexible heterogeneity versus mass heterogeneity. So the first system is Anaphase promoting complex, or APC. It's 1.2 megadalton scaffold with 19 core subunits, and it's purified from insect cell where there is both the full complex and its subcomplexes, and there are several subcomplexes. And the goal in the purification is to enrich the full complex. So when you take it to your structural studies, specifically with cryo-EM, you will have the maximum number of full complex to average out. So it's 3-step purification, and it includes strep-tactin affinity chromatography, and then you have ion exchange chromatography, and at the end you have SEC, which is size exclusion chromatography. And here are two examples of both the chromatograph signal and gels, and you can see both in the ion exchange chromatography and in the size exclusion. Here are the selected elution and their corresponding gels, that the signal is not informative. So you can, of course, get information about the sub-units that are present, but you cannot quantify how much of the full complex do you have and how much it's subcomplexes. So we took exactly those samples and measured them with mass photometry. And here are the mass histograms here for the different elutions in the different steps. And already you can see clearly that the full complex and its abundance already changes from sample to sample. And just by looking at this, you can say roughly which sample is the best sample to take to your cryo-EM studies. We wanted to do it also in a quantitative way. So we quantify how much of the full complex you have in each sample. And this is this graph here, where the circles is the quantification of mass photometry. Now the question was, is it true? Is it really true what you have in the sample? And in order to verify it, we did also negative staining EM. So we took again exactly those samples and stained them and took them to an EM facility and record a lot of micrographs and then average them. And here you can see the 4 most populated 2D class averages for all the samples, where asterisks correspond to a full complex 2D representation. And if you compare the quantification that we did in mass photometry and in negative staining, they agree in an excellent way. The only difference is that for us it took maybe an hour to quantify it, and for in negative staining it took weeks. So just to emphasize, we detect the known complex and its known subcomplexes. The quantification of the APC agrees well with negative staining, and it's a really quick and simple tool to prioritize and optimize your protocols. The nice thing about webinars is that you can show also data which eventually didn't get to the publication step. So also one thing that we wanted to see is if we can follow the stability of the protein. So sometimes you do experiment and then you keep your protein on ice, and then you want to come the next day and you want to make sure that the protein is really in the same quality as it was the day before, maybe the week before. So what we did is we took a sample, we kept it at high concentration on ice, and we diluted every day. So we did it for several days. Here you can see day 1, day 2, day 3, and day 7. And here you can see the full complex, and if you keep it at high concentrations, it's quite stable for at least a week. We wanted to see also if this also applies to diluted samples. So from day 2, we kept a sample in diluted state where we kept it on ice. Throughout the rest of the week, what we got is disassembly of the full complex. So really, it's again a really nice tool to follow up on stability. The second system that we investigated was Proteasome. Proteasome is a complex that consists of two subcomplexes, 20S and 19S. And here you can see that if you take 20S and you combine 20S and 19S, you get 26S. And if you join another 19S, you will get the 30S, which is the full proteasome complex. This purification is endogenous from specifically bovine heart tissues, and each prep provides you with different ratios between the 26S, the 30S, and also the 20S. Since it's an endogenous purification, you also co-purified several cofactors. Specifically, what we are talking about is ECM29, where it's a cofactor that helps with the connection of 20S and 19S. And here you can see a nice micrograph of proteasome, where you can see the different subcomplexes. And here are 2D class averages of either 20S in different configuration, and the 19S, the 26S, and 30. And if we look at the gels, here you can see a lot of bands, but specifically to quantify exactly what you have in your sample is quite hard from this gel. And here are two examples of mass photometry, mass histograms that we took with mass photometry of proteasome in different preparation. And the nice thing is that you can already, just by looking at the mass histogram, you can already get information about what you have in your sample. And you can see quite nicely that the two preparations are quite different, specifically with the cofactors that was purified in the sample. So here we wanted to show how mass photometry can be used to screen buffer conditions. And specifically what we did is we quantify the disassembly in different buffer conditions, either in high salt, which is in this case, and I will show you in a minute in a different condition. Here we increase the Na, the salt concentration between 0 and 500 millimolar in the buffer, and we followed the mole fraction of each one of the species. And we compared it to negative staining EM. So first you can see the mass histograms, and you can already see quite nicely how the complex disassembles once you increase the salt. This is happening specifically above 100 millimolar, where you can see the 30S completely disappears, and then above 250 also the 26S disassembles. We quantify it both in negative staining and in mass photometry, and you can see the results fit or agree quite nicely. The nice thing is that, as you can see here, we could easily detect the complex with the cofactor with ECM29, both with 26S and 30S. However, in negative staining, it's hard to distinguish between the two species, between the 26S and the 26X with ECM29, or the 30S with 30S ECM29, and therefore they could not distinguish between the two. And here you can see quite nicely that we can distinguish, and we can quantify and study the mechanism of disassembly. And you can see that first what this disassembles is the complex with the ECM29, and only afterwards the complex without it disassembles. And this fits what we already know in literature. So here mass photometry provides you with added information that cannot be detected, or it's hard to detect it in negative staining EM. As I promised, also we did it with another buffer condition where we changed the nucleotide present, and we followed the mass distribution of the proteasome. Here it's a different prep, and as a result of the different prep, the starting point is different, which is the control, the mass histogram here, the control at ATP. What we did is added hexokinase, basically changing the ATP to ADP. And we saw that there was not a significant change, and this is expected as ADP should not cause disassembly. However, when we added Aperase, which is changing the ATP to AMP, you could really see that both the 26S and the 30S disassembles completely. The nice thing is that when we added inhibitor epoximycin, we could see that the disassembly was at least partially prevented. Here it's on the top mass histogram. So again, a really simple tool to screen for different buffer conditions. The third system is nuclear pore complex, where we wanted to explore how mass photometry can help cross-linking protocol optimization. It's known that in order to make sure that the full complex is in high abundance when you dilute the sample, you can cross-link it. The question is, what is the best cross-linking protocol? So you can either play with the amount of cross-linker that you add, you can play with the time that you leave it to cross-link before you quench it. And the main question is how you quantify really each one of those options. Of course, ideally you want to do it in a high throughput manner, otherwise it will be too complicated to optimize it. So specifically with the nuclear pore complex, there are two complexes. This is complex 1 and this is complex 2, and you combine the two, and then you get the full, what we call nuclear pore complex 3. And it dissociate with dilution. So we wanted to create a quick tool to optimize this protocol. And the nice thing here you can see below is without any cross-linking, and you see the full complex here. which is in relatively low abundance. So if we cross-link it at, of course, high concentrations, and then measure it after a certain time, here either 5, 10, 15, and 20, we could see high abundance of the full complex. And we could see that already after 5 minutes, you get the maximum abundance of your full complex. So there is no really point in waiting more than 5 minutes. So we wanted to show that this is really reproducible. So we took both uncross-linked sample and also cross-linked after 5 minutes, and we repeated several times between 7 and 9. And you can see on the right that it's highly reproducible. Also, the nice thing is that you can see a shift in mass of the cross-linked protein, and the reason it's related to the added mass of the cross-linking agent. The fourth system is Respiratory Complex I, which is a membrane protein. So this Respiratory Complex I is a membrane protein with both parts, which is transmembrane domain, including 7 subunits, and also a soluble arm, which includes 6 soluble subunits. And it's known that its soluble arm disassembles, at least partially, and also disassembles through time. And here we quantify, and you can see really nicely that you have the mass histogram versus either counts or probability, where you can quantify the full complex with the transmembrane and the soluble arm, and also its partial soluble arm or its full soluble arm without the transmembrane part. And you can really assign each one of those peaks, and you can see exactly what you see. To the right, you can see the micrograph and the assigned subcomplexes or full complex, which correlates to exactly those species. So here, the important thing is that you can quantify it also for membrane protein, and you can do it in a quantitative way. Another thing which is really nice, which is also related to stability check, is that some proteins are not stable even at minus 80. As I told you, this protein is known to disassembly, specifically the soluble arm. So it really depends on how much time passed since the purification. And here I wanted to show you this mass histogram was taken approximately 6 days after purification, where it was kept in minus 80. And we also measure it after 6 months. And you can see here really clearly that the full complex, which is here, its abundance decreases quite substantially, while the other amounts of soluble arm or the partial subcomplex increases. And you can again quantify it. You can make sure that your protein is exactly in the condition that you want, or at least you have a check of your starting point when you start your experiment. And these results are related to paper that we submitted. It's in bioRxiv, and it's discussing only membrane protein in different mimetic systems. And if you do membrane protein, I encourage you to look at this bioRxiv and see how mass photometry can help you with your membrane protein. So the last protein was Cohesin, and here it's the case of flexible heterogeneity. So Cohesin is a floppy protein, and it's responsible for packing DNA, and it exhibits conformational heterogeneity. And here is an example of a negative staining image of Cohesin, and you can see it's really hard to see anything. And the reason for that is because of the different conformations of Cohesin. So you have to take a different approach in order to see it in the EM, and the different approach is rotary shadowing. And here you can see that you can detect the protein, and you can detect it in different conformations. This is really nice, but it will not give you any information about the mass heterogeneity of your sample. So you don't know if you have the full complex or the subcomplexes just by looking at the micrograph or doing any image analysis on it. Now, if you look at mass photometry, and here is the mass photometry of the same sample, you can detect that you have a really excellent sample. So mass photometry can quantify your mass heterogeneity without taking into account the structural heterogeneity. And the reason for that, and of course this is only for proteins which are at length scale which are sub-diffraction. And the reason for that there is apparent insensitivity of mass photometry signals to large-scale conformations, meaning that at least the changes in the signal due to structural conformations are within the anyway noise that we have in the measurement. So this is really nice, and it can help you with quantifying your sample when you have structural heterogeneities. So if we conclude, I hope and convince you that mass photometry is a really excellent tool to quantify your protein samples. It provides with quantitative information on sample composition. Its characterization agrees well with negative staining EM results. It can help you with optimize and prioritize your samples. And the key advantages are its native conditions, it's extremely fast, it requires minimal sample, you don't need any prior knowledge on your protein structure, you can get direct information on sub-assemblies, and you can characterize species that are difficult to visualize by negative staining EM. Of course, there are limitations. You don't get any information about structural details. And there is a limited concentration range, so you must be below 100 nanomolar of objects. However, this is something that is work in progress, and we are working now to increase the concentration range in mass photometry. Just to acknowledge, this work was successful due to many collaborations, specifically in Oxford University, Philip Kokura group. Here is our recent photo, and specifically Philip and Gavin contributed a lot to this work. In IMP in Vienna, both David and Katerina were a significant part of this study, where Katerina did all the EM of Proteasome, and also the Peters lab. And in University of North Carolina, Tanya and Nicholas were also a significant part of this work, where Tanya did all the EM work on APC. In MIT, Thomas Schwartz and his postdoc, and they were responsible for the nuclear pore complex. And in Freiburg University, Johannes and Thorsten, where they were responsible for respiratory complex I. I want to thank you for your attention and for staying tuned until now. As was mentioned in the introduction, I recently joined Refine Ltd as field application scientist. I'm based in Israel, and if you want to contact me, please, here is the email, and I would be happy to take your questions now.